Good morning, ISMB. My name is Eric Kernfeld. I'm going to talk today about benchmarking computational methods that predict gene expression following genetic perturbations such as overexpression or knockout experiments. There's tons of reasons to be interested in expression forecasting. It could be used to follow up on genome-wide association studies, figure out which genes in a given locus are causal, and which cell types are more relevant to a given association. Expression forecasting could be used to determine which genetic perturbations would move a diseased cell type closer to a healthier state. This could be used as an initial step in drug screening. Expression forecasting can be used to optimize stem cell protocols by figuring out what changes on a molecular level will coax cells towards a desired fate. And expression forecasting also has basic science value as a way to test computer models of cells. There's a rich variety of expression forecasting methods, and more of them are coming out right now. These methods span all the way from conventional structural equation models to transformer architectures pre-trained on tens of millions of cells. And they use diverse sources of prior knowledge, including transcription factor binding motif analysis and functional annotations from the gene ontology. In order to realize the potential of expression forecasting for basic science and translational applications, we need to understand how accurate it is, what approaches work best, and where are the limits of our various sources of data and contextual knowledge. In order to begin answering these questions, we have assembled and quality controlled a collection of publicly available perturbation transcriptomics data. This consists of Perturb-seq and similar experiments so that we can train up various models and then in very fine-grained detail, transcript by transcript if necessary, we can assess how well the predictions reflect the true outcome of each perturbation. Here on this slide, you're looking at the log fold change of the targeted gene on the x-axis, and then on the y-axis, you either see the frequency of that effect size, or you see a different type of effect size measured across all genes, not just the one that was targeted in that specific sample. As you can see, the overexpression and knockdown work quite well at affecting their target genes. The knockouts also generally decrease the expression level, although it is possible to have persistence or even increase in the transcript levels, even when the coding sequence has been altered so that the gene product doesn't function anymore. And then on the right, you can see that these are mostly small effects globally, except in certain cases like the Norman data, which is selected for the most dramatic results based on a low throughput CRISPR screen. What these data enable us to do is a very specific type of train-test data split. Rather than splitting the data using a simple random sample, we can select one set of genetic perturbations for training and then a completely disjoint set of genetic perturbations to test on. So we are testing counterfactual predictions here. One of the simplest ways you can get this type of prediction is to train a regression model to predict each gene from all of the other genes' expression levels. You can initialize the model with expression averaged across the control samples. And then if there's a gene that's knocked out, you can set that to 0. Or if there's a gene that's overexpressed, you can look at the test set and set that gene's value to its level after perturbation. And then the idea is to test how well the model is able to propagate that effect when you go to predict expression of all these other genes using your regression models. So we used this type of experiment to compare 9 different methods from scikit-learn. I was excited to find out what works best, except that actually taking the mean or the median of the training data always works best across all of these data sets. So we spent a lot of time going into the literature and looking for various tactics that we could implement or methods already published that would help outperform these trivial baselines. And one of the most consistently successful methods was called GEARS, or Graph Enhanced Activation and Repression Screener. We ran the same type of experiment, and we found that GEARS is not able to outperform baselines when we use traditional metrics like mean squared error or mean absolute error, or the Spearman correlation between the observed and predicted log fold change. But GEARS is generally able to outperform baselines when we look at the mean squared error on the most affected genes, or when we look at accuracy in assigning labels to clusters of cells. The exception is that GEARS is designed for single-cell data, and for data where we have only bulk RNA, or where the single-cell data were too large to ship with our software, we haven't yet found a way to get GEARS to outperform baselines on those data sets. We've done a lot more work than what's shown in this brief talk. We're planning even more in the near future. We have some fantastic infrastructure set up with tons of flexibility to explore different methods, different data sets, user-provided networks, user-provided performance metrics, and new experimental layouts. We even have the ability to split the data in different ways so that we can benchmark different tasks, such as demultiplexing of compressed perturb-seq data. We'd love to hear from you. You can check out our code base using this QR code. And finally, I just want to thank my co-authors at Johns Hopkins Biomedical Engineering, and I'm grateful to Roman Lopez, Yusuf Rouhani, and Christina Theodoris for help using their methods. Thanks for listening.